With that, I would like to introduce and welcome the final speaker of today, last but not at all least, Jared Westbrook. So Jared's going to be talking with us today about American chestnut and some updates with that. Dr. Westbrook completed his PhD in plant molecular and cellular biology at the University of Florida in 2014. After this, he started with the American Chestnut Foundation in 2015 and became the Director of Science for the organization in 2017. His primary research focus at the American Chestnut Foundation has been to apply genomics to all aspects of chestnut restoration. And you all are working on a lot of chestnut restoration from a lot of different perspectives, right? So that's really exciting. And I know that this has been a big year for you all, a lot of things happening. So really appreciate you joining with us and talking about this issue, you know, American chestnut and chestnut blight, which sometimes gets referred to as a historic disease, but it's here, it's present right now, and it's impacting things right now. And so I think look forward to hearing about the work you all are doing to change that in the future.
Thanks for having me. So today I'm going to talk about the first 100 years of restoring the American chestnut, and I think this is going to take a long-term effort to get this tree back to its 200 million acre range. So there's a lot of work to be done. So many of you know the story of American chestnut. It used to be a dominant canopy tree in the Appalachian Mountains, eastern Kentucky. It was very, very abundant. You can go around all through like Appalachia, see these old barns with chestnut wood in it, wormy chestnut. It's very highly rot resistant. It was a keystone species for wildlife. As the chestnut declined, it's thought that wildlife carrying capacity of these forests also declined. So the story is like in 1904, there was plant trade between Asia and the US, and brought over this fungal pathogen where the Asian chestnut species, it lived on Asian chestnut species, but didn't cause a disease. But the American chestnut species was a naive host in it. And it's a chestnut blight. It's called Cryphonectria parasitica. It infects the stem of the trees, and then it girdles the stem, and then the tree can't transport water up to the canopy. So the tree dies from the crown down, but it's able to resprout. So by 1950, the blight had spread throughout the original range of the species, going from Maine all the way down into northern Mississippi, and it decimated about 4 billion trees. So you can see this is a picture of Shenandoah, areas where chestnut used to be highly locally abundant. And you can see that it's like a moonscape of death here. And so today the trees live as a shadow of their former selves. They're living in the forest understory. They resprout from the roots, and then they die back in a Sisyphusian-like cycle. They're no longer able to reproduce on their own usually. So they'll get up, maybe a few of them will flower, but they will be not near a neighboring tree that's flowering. So the tree is basically frozen in time, no longer evolving on its own. So the American Chestnut Foundation, we are pursuing what we call the 3 burr strategy, which includes breeding, there's biotechnology, and biocontrol to restore the species. And the 3 burr, there's 3 nuts in a chestnut burr, so that's where we get this our acronym. So the breeding part, we've been breeding Asian chestnut species that are resistant to chestnut blight, but they tend to be shorter in stature. They've been bred for millennia as nut trees. And so that they tend to be shorter in stature, and they don't grow to the canopy. With American chestnut, which is a dominant canopy tree, but is not resistant to blight. So there were some early breeding programs to do this, starting in the 1920s even. and going through the 1960s to make a lot of crosses between these Asian species and the American chestnuts. And basically we got trees that were intermediate in resistance and not super competitive in the forest. So in 1983, the American Chestnut Foundation was founded based on this idea of doing back crossing, where we take the 50-50 hybrids between Chinese chestnut and American chestnut, and we back cross it to American chestnut, inoculate the trees with the chestnut blight every generation, and select individuals that inherited the resistance from the Chinese trees, but to have more American characteristics. So that's the purpose of back crossing, is to get trees that grow more like American chestnut, but they have the resistance from Chinese chestnut. And this idea is based on the assumption, the hypothesis that the blight resistance is fairly simply inherited trait, that at every generation you can inoculate a tree, you can pick out the trees that inherit the resistance and move forward in that way. So you're not really diluting out the resistance. And now we've gotten to the stage where we're taking these third back cross trees, which are like 95% American chestnut, crossing them together, and then generating these large populations where the trees have a chance of inheriting resistance from both parents. So it enables us to enhance the resistance. So we've been working with a network of citizen scientist volunteers. There's a chapter in Kentucky. If you go to TACS website, you can learn how to join that chapter. And basically what the chapters have done is we've sent people pollen, and they've actually contacted their local utilities and found flowering trees. A lot of people just like find these flowering trees, and then they pollinate the wild trees that are flowering, that happen to be, you know, kind of on the side of the road or something, that they flower before they die back from blight. And then we're able to capture that germplasm by way of breeding our hybrids with these wild type American chestnuts. And you can see this map of where we've found trees throughout the range. So we have some in eastern Kentucky, into the land of the lakes. So we're still always looking for new American chestnuts to bring into our breeding program to diversify it. So now we're at this final stage of breeding, and we planted out 60,000 in our Meadowview research farms, which is in Southwest Virginia, which are our flagship research farm. We planted 60,000 trees from these final, these advanced generations. And when we inoculate them with the chestnut blight, it's like a roll of the dice, whether a tree inherits the genes for resistance or not. So you see these backcross trees demonstrate a spectrum of blight resistance when we inoculate them with a blight, with very severe reactions to trees that have minimal cankers that are healing. And so we're basically aiming to cut down all but 1% of the trees. So it's a lot of work. That's why it's taken so long, is just planting out so many trees and sifting through and finding the best ones. So I just wanted to kind of show you what, after this, we've been doing this for 37 years, and it's been an experiment the whole time. So we want to know what's happened based on some of these assumptions about the genetics of resistance. What have we actually come up with in the end? And you can see we have trees in the seed orchard. So there's an American chestnut. You can see that it grows and dies and gets the blight and resprouts over and over again. So obviously that tree doesn't have any resistance. When you get trees that have some Chinese genes for resistance, you end up getting partial resistance to the chestnut blight. So the trees are not as resistant as Chinese chestnut like we had hoped. But they do survive with the blight. And some of these trees, this tree was planted in 2003, for example, it inherited 93% of its genome from American chestnut. It's got cankers up and down the stem, but it's still hanging in there. And you can see in our orchards, we have this whole spectrum of Chinese and American chestnut inheritance. So as the trees inherited more of their genome from Chinese chestnut, they tend to have higher levels of resistance. That's one of the things we've noticed as we started doing DNA testing on these trees. we found that some of our trees even were outcrosses to Chinese chestnut, meaning we had these backcross trees that were 95% American chestnut. They accidentally got pollinated by Chinese chestnuts nearby, and those trees ended up being the most resistant in the orchard. So we have this kind of intermediate level of resistance and intermediate level of American chestnut characteristics right now, from after doing all this calling and selection. So it's been partially successful in the breeding. And we're still continuing to cross the best trees together and move forward with a traditional breeding program. But we also have this other approach that we're using, biotechnology, where we've put in, our collaborators at the State University of New York have put in a gene from wheat that detoxifies an acid that's produced by the fungus that creates these cankers on the trees. It's called the oxalate oxidase gene, and it basically turns that acid into byproducts that are non-toxic to the tree, carbon dioxide and hydrogen peroxide. A lot of people ask, is this gene like allergenic, like because it's from wheat, is it going to create gluten intolerance or something? It has nothing to do with gluten. This gene is present in a lot of things that we eat every day: bananas, rice, barley, sorghum. And you can see these pictures of wild type American chestnuts that were inoculated with the blight. They're wilting, and these darling trees, we call them darlings for one of our volunteers that funded some of this research for a long time. These trees are standing up to the blight, so they definitely have some resistance, and it's advantageous to use this because it's a single gene, so it's easy to breed into a lot of different genetic backgrounds. So there's been some research done on, is there any unintended negative ecological consequences from inserting this gene into American chestnut? Does it affect other things besides blight resistance? And so the researchers at SUNY have put the leaf litter, they've tested, tried to germinate seeds from other plants in the leaf litter. They've compared the herbivory on the leaves of the transgenic trees and the wild type trees. They've compared the colonization of roots by mycorrhizal fungi, bees feeding on the pollen of chestnut, tadpoles feeding on the leaf litter. So they've done a lot of different experiments and basically have shown no difference between the transgenic and the non-transgenic trees in terms of these other non-target ecological effects. Of course, they've also looked at the nutrition of the nuts, and basically there's no difference in the nutrition of the nuts. There might be some difference between individual trees or between European chestnuts and American chestnuts and Chinese chestnuts, but this gene has no effect on the nutrition. So there has been this review process. There's been the first public comment period on it in October and of last year, and so we're still waiting on, and it might be a year or two yet before we have a decision from the USDA as to whether we can release these trees. So we're, in the meantime, we've been doing some crossing between these with permits from the USDA. We've been crossing these trees to a number of different wild type trees and some of our hybrids. And working with these trees has an advantage in that we can, rather than calling through huge numbers of trees in seed orchards, we can actually just take a nut from one of these crosses, pull out a little core, put it in a solution. If the tree inherited the gene, the solution turns black. If it didn't, it's clear. So it's an easy way to do a selection. And then once we can grow these seedlings out and put them under high light, and chestnuts flower during the longest days of the year. So basically you can force these plants to flower as seedlings. So it enables us to do multiple generations of crossing relatively quickly. So it has a number of advantages logistically to be working with these trees. And the big question is how how resistant are they? So we're also doing some plantings with these trees and just making sure over the long term that the resistance of these trees holds up. And we really need the regulatory approval to be able to actually just do the basic research and allow these trees to grow up and flower and everything in the field in order to assess their long-term resistance. So one way that you can help us out now is finding American chestnuts and volunteering with the chapter in Kentucky. And we've been working with Ellen on using tree snap to find American chestnuts and document where they are. And one of our goals is to conserve 1,000 different wild type American chestnuts from across the range. So what tree snap helps us do is track the locations of all those trees. And this winter, we're going to a number of different locations, including in Kentucky, and going back to the American chestnuts and harvesting scion wood so we can graft the trees and propagate them in orchards where we can use them for future breeding. Of course, if you find a flowering wild type American chestnut, great. That's what we really want to see. And if you can harvest nuts, wonderful. But those are very rare. Another thing is we're working with a researcher at the University of Maryland who's engineered the chestnut blight fungus too. And this fungus is able to be infected by viruses that make it less virulent on American chestnut. And this has been used effectively in Europe to control the spread of chestnut blight. But in the US, there's a lot higher diversity of the chestnut blight fungus, and the strains of the fungus need to be genetically compatible to transmit these viruses. So what this engineered strain of the Cryphonectria fungus does is it enables transmission of this virus to many different diverse strains of the fungus. The issue is you need to treat every single canker, and there's millions of trees, so this can't be possibly effective for landscape scale restoration. However, keeping trees alive in an orchard is very useful for crossing the wild type trees to the darling trees or hybrids. Finally, chestnut blight is not the only disease that affects chestnut. There's another disease that was imported from Asia that you might know about called Phytophthoracinnamomi. It causes Phytophthora root rot. It kills thousands of plant species. American chestnut happens to be one of the most susceptible. And it turns out that the Chinese chestnuts are resistant to this disease. And some of our hybrids inherited some of that resistance from Chinese chestnut. We've been screening thousands of our hybrids every year for resistance to the disease in collaboration with the U.S. Forest Service in Asheville. And some of the trees have these robust root systems even in the presence of this disease. So we are crossing these darling trees, the blight-resistant trees, with our hybrids that have resistance to the Phytophthora to get both types of resistance. So we can basically have two types of resistance to the major diseases that affected American chestnut. So what are we looking for in the end? We want the trees to look like American chestnut, grow like American chestnut. We want the trees to survive with blight indefinitely. Some of the population should have resistance to the second pathogen. And then, of course, we want to bring in enough genetic diversity so that the tree can adapt to a wide climatic range and to climate change. So I think, with all of the technology that we have available to us now, that it's imminent that we are being successful doing all of that with crossing the darling trees, bringing our Phytophthora resistance, doing some germplasm conservation on a wide range of individuals from all across the range. So we definitely still need people's help, though, in terms of finding the trees and helping us with the germplasm conservation. So, one of our board members wanted to have a tagline for us that it's more than just a tree. It's a community of people that have spent their decades working on this, have made a lot of friends. And so a lot of people that have retired, they've been helping us out managing the orchards. It's amazing. We have thousands of volunteers in 23 states that help us maintain these orchards. We were recently highlighted by a panel by the National Academy of Sciences, addressing the issue of using biotechnology for conservation, which is something that's unconventional, and that it could be applied for species like the emerald, for the ash trees, for example. So chestnut has been around, this problem has been around for the last 50 to 100 years. So we've had kind of some time to work out some various methods. So, but I imagine this, these different biotechnology could be used for other tree species. And then we found recently bees, a rare bee that hadn't been spotted for, 50 years, growing on, that was feeding on chestnuts in Connecticut. So restoring the species could have some positive effects on specialist pollinators, for example. So how can you get involved? We have a chestnut chat series every month. So we have different speakers talking about different aspects of chestnut restoration. We're a nonprofit member organization. It costs $40 to become a member. And then we have Chestnut Magazine. You can learn all about the latest developments in chestnut restoration. And of course, download TreeSnap, learn how to identify American chestnuts and document the locations. And that really helps us. And of course, volunteer with your Kentucky chapter. And with that, I'll take any questions.
Well, thank you so much. I think we do have a few questions in the chat. If you'd like to stop sharing your screen, you could open that up and check out what's been placed there for you. We have one about the canopies of the back cross breeding. How do they compare to the pure American chestnut? And wasn't that one of the best qualities of American chestnut?
Let's see, where is the
Q&A is kind of at the bottom next to the raise hand section.
Okay. So the backcross trees, in terms of like, what is I don't know exactly what you mean by the canopies, but the trees that we've selected have lived with their main stem alive for 15 to 20 years now. And they inherited, 85% of their genome from American chestnut, for example. So they do seem to be, their stem is surviving relative to American chestnuts, where the main stem gets the blight and dies over and over again. So in terms of like, will they grow up to being a canopy tree? It's difficult to assess that. We have a number of tests in the forests, like the National Forest System, where we do have trees that have been living for like a decade or more, but it takes a long time for these trees to grow, and to assess that question, like, will they grow up and be competitive in the forest? What will be the maximum height growth that they achieve? Those are long-term, those questions require long-term studies. So we are putting out some of these long-term studies to test the forest competitive ability.
So one of the questions there for you that I think you'll be able to speak on is why can't we use, or can we use, genetic engineering to transfer the Chinese resistance over to the American trees? That was part of the goal of the backcross program, and now that we have that technology, is that possible? And why or why not?
Yeah, we're sequencing the genomes of Chinese chestnut and American chestnut, and we're mapping the resistance, where the resistance resides. But to actually identify the individual genes is very challenging. And then to put them into American chestnut in the right combination is also sort of unprecedented. I can't even think of an example in systems that are easier to work with, like corn, for example, where there's a complex trait and controlled by many genes, which seems to be the case for chestnut blight resistance, where they've engineered that into a plant that's easier to work with, and then been able to measure the effect. So I think we are doing some of that research to understand the genetic mechanisms of resistance, but it's still going to be challenging to find the right combination of genes and expressed in just the right way at just the right time to lead to high levels of resistance. So we're hoping that the OXO gene from wheat does have a strong effect on resistance. We're also breeding that in with our backcross trees that have some resistance from Chinese chestnut. We don't need to know the genes to know that our trees have partial resistance, and we can select for it. So we're trying all of the above. So I see, are there any mature American chestnuts anywhere? There are maybe 10 or 20 individuals that survived the blight that I know of that are still living, and curiously, they have partial resistance. It's not enough to restore the whole species, starting with those trees. There are American chestnuts on the West Coast. They're planted and they're blight free. There's American chestnuts, and I know the largest one I think is in Belgium. So outside of where chestnut blight is, yes, there are mature American chestnuts, and there's probably about 400 million American chestnuts estimated to be left out of 4 billion trees that were originally here. And they're growing as little root sprouts, and some of them are tiny or seedlings that have never even gotten the blight. They've been probably suppressed the entire time. So there's a lot of American chestnuts left on the landscape. They just don't become competitive forest trees. In the orchards, when removing blighted trees, do you wait for blight to completely? What we do is for assessing resistance, we inoculate with chestnut blight, and then we wait like a year. And then we've also been assessing the long-term resistance over a number of years after. So when we make the final selections, we use, resistance phenotypes on trees that have been inoculated for 5+ years. We use the genomics to help us make more accurate selections. So yeah, we try to look at the long-term resistance of the trees.